Okay, so now the second part, acid-base balance. So basically we want to talk about buffers. Now in our bodies we have got enzymes. Enzymes are basically proteins, and these proteins work in a certain pH, just like in a certain temperature. And these enzymes will work well at an optimum temperature and at an optimum pH. Meaning if you change the pH very much, those enzymes will stop to work. Most of the enzymes in living cells will maintain a pH of 7. They will be like most of this pH in the cell, most of the operations are between the pH of 6 and 8. That is like 7 average. So any fluctuation, any change in pH will affect those enzymes. So that means you need to have a compound such that, let's say to a cell which has got a pH of 7, you add an acid. What will happen is that if you add an acid or if you add hydrogens, the pH will be dropping, it will be becoming more acidic. So you want to have something that will make sure that even if you add an acid, it will be able to fight that acid to maintain the pH. Even if you add a base, to be able to fight that base to maintain the pH. So that is what we call a buffer. So buffers are very important because they'll be able to maintain pH. So they will resist changes in pH. Now, they will not resist big changes in pH. They actually just resist small changes in pH. A narrow change in pH. Physiological buffers of importance which we describe are bicarbonate buffers, phosphate buffers, and protein buffers. And we should know that for a buffer, hydrogens, when it accepts hydrogens, it becomes acidic. Like hydrogens are negatively charged. I mean, as hydrogens would be the ones which make something to become acidic. So every time it accepts hydrogen, it becomes acidic. When it donates hydrogen, it becomes basic. Or when it accepts hydroxide, it becomes basic. So that's what we need to take note of there. Buffers will act as a first line of defense against changes in pH, just like we have got the innate immune system. They are actually part of the innate immune system. They will act to maintain, to fight anything that would want to change the pH. Most of the fluids in the cytoplasm of cells contain high concentration of proteins, some organic substrates, inorganic salts, and all of these will be acting as buffers to maintain or to resist change in pH. You also have nucleotides like ATP. ATP and many other low molecular weight metabolites can contribute to buffering capacity of the cell. Buffers are going to be biologically important if the pH value of blood remains in either alkaline or, okay, it wants to change from alkaline to being basic or the other way around. So buffers will be important because immediately the pH changes, that can be harmful to the body. That is because biochemical reactions will happen within a given pH, and that might prevent the working of some organs if the pH changes very much. So that's why buffers will be very necessary. So talking about these buffer systems, buffering is an important concept when we are talking about living systems in maintaining constant internal environment, and that is known as homeostasis. Small molecules like bicarbonates and phosphates will provide buffering capacity. Same with other substances like hemoglobin and other proteins. The major buffer systems that we'll be talking about is the carbonic acid bicarbonate buffer system, the phosphate buffer system, and protein buffer system. Just these three. Starting with the carbonic acid bicarbonate. Now, when you're talking about carbonic acid, is this one, H2CO3. Now, what will be the conjugate base of this acid? Remember, the conjugate base is formed when this acid loses hydrogen. So it will have hydrogen plus HCO3 with a minus, right? We said anything, every time something loses hydrogen becomes negatively charged. Now remember, we said that when you want to maintain this, you don't want it to be in the way it is negatively charged. You want to make it neutral. So you can use group 1 elements like potassium or sodium. So what you do is that you are going to attach sodium in front. So it becomes sodium and then hydrogen carbonate. So this is the conjugate base of the acid carbonic acid. So these are the components of the carbonic acid bicarbonate. So this is carbonic acid, this is the bicarbonate buffer system. So what will happen is this: when we add an acid to our solution, so let me ask, between this one A and this one B, which one is the base here and which one is acid? Which one is the base and which one is acid? A or B? Okay. Okay, good. So the acid is always the one which is having more hydrogens than the other one. So what will happen is this: if I introduce an acid to this buffer, what will happen is that the basic component of this buffer will be the one to react, and the base is B. So if I react an acid, and you know the characteristics of an acid, let's say for example I introduce hydrochloric acid in this buffer. So that hydrochloric acid will be welcomed by the sodium hydrogen carbonate because it's a basic component. And what will happen is that, remember, this sodium is positive, right? The chlorine is the one which is negative. So the sodium and the chlorine will react to form sodium chloride, which is salt. Okay, and then the hydrogen here will react with the bicarbonate which remains to form carbonic acid. So if I add an acid to my buffer, we are forming more of the acid. Okay, so that is what is happening. At least we are forming this kind of acid, and this acid can still act as what? Can still act as a buffer because it is part of the buffer, which is this one here, A. Now if I added the base, if I added the base to my buffer, what will come it is the acid component. Now the a base, let's say the characteristic of a base is having a hydroxide. Let's say for example we're reacting with sodium hydroxide. So what will come this base is the acid component of this buffer, which is the H2CO3. And here sodium is positive, it will react with the bicarbonate, which is negative. The bicarbonate is supposed to have an hydrogen and then CO3. So what will happen is that sodium will go and react with the bicarbonate. Bicarbonate is HCO3. And then the hydroxide which remains here will react with that hydrogen which came from this to form water. So you see, we have formed more bicarbonate after we added the base, and we say the bicarbonate is the basic component of this buffer. So we are forming more base. Does this make sense? Is this okay? Okay. So you need to take note of which one is reacting with which, because those are the questions that they'll be able to ask. Normally in the body, metabolism will produce acid. So because metabolism produces acid, meaning that you're supposed to have more of the bicarbonate. And this bicarbonate can be produced. Remember carbonic anhydrase. What happens is that when you have got a lot of carbon dioxide in the body, it can react with water, and then that produces bicarbonate. And then this bicarbonate can break also to hydrogen ions and then carbonic acid. So this hydrogen will be taken by hemoglobin. Okay, hydrogen 2,3 bisphosphoglycerate and also oxygen, carbon dioxide. Those can bind to hemoglobin. So the hydrogen, 40% of hydrogen is actually transported by hemoglobin, and that's an exam question. And then the bicarbonate which remains will act as a buffer because mostly the body will produce acid. Okay. So this is what I'm just from describing. The bicarbonate we said is regulated by sodium. That's why we don't just write HCO 3 minus. Instead, we put a sodium in front and then the positive is no longer there. I've already talked about what we can see here. When we are adding an acid to our buffer, it is the basic component to which you react. So you end up forming an acid and a salt. If we react with a base, it is the acidic component which will react, and so we end up forming a base and water. Now for this buffer, as we already said, the bicarbonate can dissociate to form carbon dioxide and water. Because the bicarbonate, look at this, when the acid reacts with this buffer, we are forming more of the acid, right? A lot of this acid which is being formed might accumulate in the body. So what happens is that it can be converted to carbon dioxide. Now this carbon dioxide is going to be expelled from the body via the respiratory system. That is why the carbonic acid bicarbonate buffer is related to the respiratory system because of this carbon dioxide that has to be expelled. The next buffer is the phosphate buffer system. The phosphate buffer system is going to operate in internal fluids of all cells. Okay, it is in the internal fluid. That's important to take note of all cells. Now, phosphate buffer system, what you have is first you have got the acid. Let's talk of phosphoric acid. Phosphoric acid is H2PO4. Okay, actually not like this. Supposed to have a 3 because the phosphate has got a valence of 3. So this is phosphoric acid. When phosphoric acid loses an hydrogen, it will become H2 and then PO4. Remember, when something loses an hydrogen, it will carry a negative sign. When this one again loses an hydrogen, it will become now H and then PO4. So can you tell me what will be its charge if it loses another hydrogen? What will be its charge? Okay. So remember, it had 3 hydrogens. We lost one hydrogen. It became -1 charge. If we lose another hydrogen, it will have now a post negative 2 charge. If we lose this other hydrogen, it will have a negative 3 charge. So the more the hydrogens you lose, the more the charge reduces. Now for a phosphate buffer, we don't use this. We don't use the phosphoric acid. We use these two. These two. Remember we said anything which differs by only one hydrogen is a conjugate. So meaning that these two guys are conjugates. Now let me ask another question. Between this one, let me label it as A, and this one, let me label it as B. Which one is going to be the acid and which one is going to be the base? Which one will be the acid? Okay, good. So A is the acid. Now, because we don't want to have this negative charge, we react this with group 1 elements. So because we only have one charge, I'll just add one sodium. So it becomes sodium dihydrogen phosphate. We are saying dihydrogen because we have got two hydrogens. For this other one, which is the base, it has got two charges. So we're going to add two sodiums. So this is disodium hydrogen phosphate. So in a phosphate buffer system, what acts as the base is disodium hydrogen phosphate. What acts as the acid is sodium dihydrogen phosphate. So what happens is that if I react this with an acid, it's a base which reacts. So imagine I bring in hydrochloric acid. So it's a base which will react, and the base is the disodium hydrogen phosphate. And what is supposed to happen is that the hydrogen which is positive will react with the base which is with the part which is negative. And remember the part which is negative is this one, biphosphate, hydrogen phosphate. So meaning the meaning of this is that for the hydrogen phosphate, the hydrogen is going to react there. I want you to make sure that you. Okay. So what is happening here is the hydrochloric acid, the hydrogen here, positive, will go and react with the what? With the positive part for this one, right? With the negative part, I mean, for this one. And the negative part is in this form. It has got one hydrogen, only one hydrogen. So the meaning of that is this. When I bring in a hydrogen to come and react here, because it already has an hydrogen, so it will now have two hydrogens, and then PO4. It had one hydrogen, it had the charge of negative 1. So if I add another hydrogen, I mean it had the charge of negative 2. So if I add another hydrogen, the charge will be now a negative 1. We don't want that negative to be there. So what will happen is that to maintain it neutral, we are going to add the sodium. And then for the basic component, if we react this with a base, let's say for example sodium hydroxide, it is the acid component that is going to react. And the acid component is in this case is the sodium dihydrogen phosphate. Now this is not the only product. We had another sodium which was removed from here that reacted the chlorine to form sodium chloride. So here what will happen is that the sodium which is positive will react with the negative part also. But in this case, the negative part has got two hydrogens. So what will form is sodium with that compound with two hydrogens with a phosphate. And then what happens? Remember we had a sodium here, meaning that this sodium was displaced. So this sodium which was displaced will actually react with the hydroxide which is negative here to form sodium hydroxide. So this is how the reaction actually gets to go. Does this make sense? Does it make sense? Okay. So so this is what you can see here. Now I'd written something else here. Okay. Let me use this same one here. When we react with an acid, can see we are just grabbing, we are removing the hydrogen from the acid, which is positive, to come and react with the negative part here. And that negative part only has, so this only has one hydrogen. So it will now have two hydrogens. And then one sodium will still be there. And then the other sodium which will be removed will react with the chlorine. Sodium hydroxide, if it reacts, the sodium which is negative, I mean positive, will react with the negative part. Remember the negative part of here is actually sodium, like that. Let me first write it without the sodium. What I'm writing is this one here. So we have got hydrogen with a phosphate. This has a charge of -1. And then we are going to react it with this sodium, which is at this point. But remember we said to maintain the charge, that's why we added that sodium. So that's why we are ending up with, okay, let's do this. Let me react it. Let me first remove this sodium. This is something that I didn't explain well. And then I've removed also this sodium. So what we have remained with here is hydroxide. So these are the two which are actually reacting. So we are going to remove one hydrogen from this guy. That hydrogen will come and react with the water, the hydroxide. That will form water. So the meaning for that is that we have actually removed one hydrogen. So it has only remained with one hydrogen because it had two. And then PO4. So it is going to have a -2 charge because it only has one hydrogen. So the two sodiums which remained will now be the ones which will come here to form back your disodium hydrogen phosphate. Now the charge will no longer be there. So the most important thing for you to know is which one will react with which one. So this is just an example to talk about calculation, just to identify which one is acid and which one is a base. This is the Anderson-Hasselbalch equation. You have sodium dihydrogen phosphate and then disodium hydrogen phosphate. So the one with few hydrogen, this is the base, and then this is the acid. So in this case, on top we have acid for the Anderson-Hasselbalch equation. pH is pK plus log of base over acid, sorry. So the base on top we are going to put this concentration, which is this one. The bottom is concentration is this one for the acid. And then that will give us a pH of 7 there. The other type of buffer is a protein buffer. So I've talked about the carbonic acid bicarbonate, the phosphate, and then the protein buffer. Protein buffer is in plasma, blood plasma, and also in cells. Amino acids which make up proteins, amino acids are in this form. You have got a carbon bonded to a nitrogen, which has got three hydrogens, so it has a positive charge. And then it is also bonded to COO minus. It has got a hydrogen, it has got also an R group. This is known as a deuterium nature. It has a positive and a negative. Because it has got also both positive and negative, it can act, because it has got both positive and negative ends, it can act as a base, also as an acid. So this part which is positive, the positive part will be the one which will be donating hydrogen. Let's say, for example, you are reacting with a base. A base has hydroxide. So one hydrogen here is going to be released. It comes and reacts with the hydroxide here, and that will form water. So the part of the amino acid that acts as a base is the amine, okay? Because the base is a proton donor. And then this uh, COO minus, which is the carboxylic acid functional group, or the carboxyl group, will be the one which will be gaining hydrogen. Let's say we are reacting this with an acid. So this acid will be able to react, this hydrogen will be able to attach there, and then you form COOH. So this one will be acting as the base. Okay. So most of the buffering capacity in the blood and also in cells will actually come from proteins. Red blood cells have got hemoglobin. Hemoglobin carries hydrogens and oxygens. So what happens is that carbon dioxide can be converted by carbonic anhydrase to bicarbonate and hydrogen, as we saw. Carbon dioxide reacting with water. First it forms bicarbonate. Bicarbonate can form, carbonic acid, and then carbonic acid can form bicarbonate and hydrogen ions. These hydrogen ions can be transported by hemoglobin. The bicarbonate can go on and act as a buffer for the carbonic acid bicarbonate buffer. So this is how proteins will be able to act as buffers. Okay. So now a very good summary. For extracellular fluid, the major buffer is a bicarbonate buffer. ECF is a bicarbonate buffer. We also have some minor buffers like proteins and phosphates. For blood, the major buffer is also bicarbonate. That is because blood is, remember, extracellular fluid, ECF, can be divided. You can have plasma, you can also have interstitium and the transcellular fluid. So both the plasma, you can see, is also part of what? ECF. That is why the major buffer is also bicarbonate. Also, you have got hemoglobin. And then inside the cells, we say the major buffer are actually proteins and also phosphates. In urine, you are going to find ammonia and phosphate buffers. So this is it.